Okay. Can you hear me okay? Someone give me a thumbs up. They can hear me. Good. My name is Hysjulien. I'm a nurse and clinical psychologist that specializes in health psychology and specifically in oncology psychology. I'm giving this presentation on behalf of myself and Dr. Cavallari, who is our numbers guy, and Amy Schmidt, who has been instrumental in this project. I also want to give due credit to all the people in research who have helped out on this. Go ahead, advance. Okay, so the project is a comprehensive approach to head and neck cancer prehabilitation. This is a project that's been ongoing in its infancy, was part of a Commission on Cancer project, and then grew into a DaCCoTA grant. And here's the final presentation minus all the results. Head and neck cancer accounts for about 3% of the malignancies in the US, and about 53,000 Americans develop head and neck cancer annually, with about 11,000 people dying from the disease. The treatment options have really advanced in head and neck cancer survivorship over the last 3 decades, and therefore we're seeing more and more survivors that around. Go ahead. Depending on the primary tumor site stage, I'm going to take a second, you guys, because there we go. Okay. Depending on the primary tumor site stage, performance status, comorbidities, and the patient's goal, treatments can consist of, and often do consist of, surgery, radiation, chemotherapy, a combination of all three or some of these modalities. They contend with treatments that really affect their quality of life. Samuels reported that treatment regularly results in decreased functional capacity and decreased quality of life, with physical impairments manifested, though not limited to, disfigurement, deconditioning, communication issues, swallowing, speech, breathing, and cancer-related fatigue. Sarcopenia, which is muscle wasting, is a really important predictor of outcomes in this patient population. It's an independent risk factor for poor disease-free and overall survival outcomes after definitive treatments, and showing and associated with a threefold increased risk of overall reoccurrence or of death. It is also highly associated with early cessation of chemotherapy and treatments, and therefore interventions based at addressing sarcopenia right from the beginning are really critical and vital to these people. Go ahead. Premorbid factors such as anxiety, depression, chemical dependency, financial barriers, lack of social support are really unique obstacles to the head and neck cancer population. Chemical dependency is big, which then affects financial issues and their lack of social support. And oftentimes people are drinking because of pre-existing anxiety and depression. So with of all the cancers, those are really uniquely high in the head and neck cancer population. Because of this, anxiety, depression, psychological distress, and ongoing fear of cancer reoccurrence are even higher in these patients than in other patients. In fact, head and neck cancer population are twice as likely to commit suicide than the general cancer population, which is already 2 times higher than the general population. They also have an extremely rigorous treatment course that really impacts them in all aspects, physical, social, and emotional functioning. Go ahead. So, much of the literature to date has really focused on functional outcomes and rehabilitation after the loss of function has occurred. So after their sarcopenia, wasting, their ability to communicate, disfigurement, depression and anxiety have already occurred, then the literature focuses on what can be helpful there. But as we know, there's a shift in healthcare in general towards a proactive approach, and that's really what we've taken on in this project, really proactively looking at and proactive approach as the basis of rehabilitation programs. Go ahead. So rehabilitation is a proactive preventative service. It describes rehabilitation as a process of on the cancer continuum between the diagnosis and the beginning of acute treatment that includes physical and psychological assessments that establish a baseline functioning level for patients and identifies impairment or risks and then provides interventions that promote physical psychological health to reduce the incidence or severity of future impairments and symptom burden. Currently, there aren't any active prehabilitation programs that we know of that have been researched for head and neck cancer patients. There have been some in other patient areas, but not ours. Go ahead. So here at Roger Mayers Cancer Center, we developed a multidisciplinary team of head and neck cancer experts, including a physical therapist, speech therapist, dietitian, integrative essentials health educator, and psych oncology. We developed a comprehensive prehabilitation clinic that evaluates each person using standardized screening tools and then provided in personalized education and interventions to maintain functional outcome, optimize well-being, and improve quality of life and during and after treatment. Also promote treatment adherence. These patients drop out of treatment frequently. So we initiated a prehabilitation clinic here in Fargo, and then we're evaluating the comprehensive approach and looking for outcomes. Go ahead. The study design included patient data will be compared using a repeated measures design as part of retrospective, non-randomized, prospective, two-armed clinical trial. I'll explain in detail in a minute, in which we assess whether prehabilitation interventions help mitigate these adverse symptoms of anxiety, depression, suicidality, distress, nutritional status, sarcopenia, dysphagia, as well as overall well-being. Go ahead. So we have prospective enrollments in a study group. We consented patients who met the criteria and participated in the prehabilitation clinic for standard of care. We attempted to enroll 40 participants. The control group was in Sioux Falls. And again, we wanted to attempt to enroll 40, but I'll get to all that in a minute. Go ahead. We also had a retrospective group, and that the first retrospective group will be part of the control group. It'll review charts from patients who've never participated in a prehabilitation clinic up to 2 years prior before the clinic starting. And then the other would be part of the study group. It would review charts from patients who participated in the prehab clinic program but weren't approached for the study, or had to participate in the prehabilitation clinic prior to the study opening. Patients previously approached or deceased obviously were eliminated, and we were going to review those as many charts as we needed to get that total N of 4. Go ahead. Inclusion criteria were patients greater than 18, diagnosis of head and neck cancer, including oropharyngeal, laryngeal, or squamous cell carcinoma, patients that were actively pursuing cancer treatment with a curative intent, and able and willing to provide a written consent. Go ahead. Exclusion criteria, as you can see, interestingly, because of the SOZO measure, included patients with cardiac arrhythmia or implanted pacemakers, electronic devices, external defibrillators, pregnant women, patients weighing over 375 pounds, and patients with metal allergies. Exclusion criteria in the prospective group were none. Go ahead. So in Fargo, the study group participated in each of the following. They obviously, after recruited in and informed consent, they participated in a 60-minute integrative essentials pre-treatment intervention. This was either administered in person or virtually, and the intervention focuses on stress management and teaches techniques for relaxation, visualization, imagery, and use of hypnotic suggestions that help prepare the person for their treatment. Patients were given exercises that they were instructed to use on a regular daily basis. Then they also met with each of the members of the interdisciplinary team in the prehab clinic, either by phone or in person, And those people included physical therapy, speech therapy, social worker, and a registered dietitian. Assessments and interventions that would be described later were administered. Then they met with each of those people in person or by phone at 6 to 8 weeks post and 5 to 6 months post-treatment. Go ahead. The assessments included a brief evaluation of the IEP, the Integrated Essentials Pre-Treatment. The physical therapist did a 2-minute walk test, 30-second sit to stand, grip strength, and measuring of functional capacity. The bioelectric impedance spectrometry, the BIS, is a measure of sarcopenia that was specifically able to be used in this study as we had the monies to buy the instrument. The nutritional focus physical exam, the NFPE, measure of nutritional status, malnutrition, and the risk of malnutrition. The MD Anderson dysphagia inventory was also used, and it identifies symptom-specific outcomes for dysphagia. The VHS-6 was used, identifying chemical dependency, risk factors, anxiety and depression. Columbia suicide risk was used as well. The FACT functional assessment of cancer therapy for the head and neck, a measure of physical well-being, social, family well-being, and emotional well-being was administered. And then the National Comprehensive Cancer Network, the NCCN distress thermometer, which is a multifactorial cancer-specific measure of distress with assessing 5 different variables and a qualitative assessment of distress. Go ahead. The prospective control group in Sioux Falls, again, did the same thing, consented, and at seen initially between diagnosis and initiation of treatment, then at 6 to 8 weeks post-treatment and 5 to 6 months post-treatment. They were administered the VHS-6 Columbia, FACT head and neck, the NCCN distress thermometer, and the MD Anderson dysphagia inventory. Go on. The retrospective study group, I keep being wishy-washy with my tense, but will be a evaluation of electronic medical records for qualifying patients, and they will be able to obtain all of this information. With the exception of the SOZO, the measure of sarcopenia is different, and it's measured by CT scan. Go ahead. In the retrospective control group, the electronic medical records for qualifying patients will be reviewed, and not all of the assessment measures will be available because they weren't used. So the BHS-6, Columbia, FACT head and neck, distress screening, and a CT measure of sarcopenia will be used. Go ahead. So I won't bore you with too much with the analysis. A repeated measures design will be utilized to monitor and assess the success of the program. Obviously, we're
2 minutes remaining.
We're looking for that response, use of ANOVA tests, and looking at multiple aspects, comparing and looking for benefits of interventions that the prehabilitation clinic did. Go ahead. Just to let you know that as part of the prehabilitation and/or the head and neck team, these interventions were monitored through the course of the study, and the team met weekly, looking and assessing at the participants' progress or detriments during their treatment course. Interventions, referrals, and modifications to interventions were done, and the nurse navigator had at least monthly check-ins with the control group patients. Go ahead. The results are not there, okay? And the results are not there because the last thing I'd just like to share with you are the multiple aspects of this study that have been extremely challenging. We will get results. They will be written up and published, but we started this study it had multiple enrollment and barriers, including a delay of a grant obtaining, and then the grant hitting right in the middle of COVID, which left us scrambling with means of administering everything that we had put into place. There was also significant changes in personnel in the study group, including surgeons that were changed and having to get redone. The SOZO machine, one of the delays was that we had to get all of the authorizations and jump through all of the different steps to be able to get approval for use of the SOZO machine, and that was not anticipated. We also, because of our recruitment, decided to do more of the retrospective analysis and include them in the study group. And then we had to find and enroll someone who would be able to obtain that retrospective data, and that's just been completed. So
If we could wrap up to have some time for questions.
with that, I'll end. Yep.
All right.
Mm-hmm. If there are any questions.
I guess I do have a question. Now, you mentioned that there are no studies that you've seen with prehabilitation with head and neck cancer. Have there been other
Prehabilitation and other cancer sites?
yeah, and other cancer
Yeah.
sites that have
Yeah.
like maybe what would some of the bigger ones or like kind of more I guess complete ones be?
Yeah. And one of the big areas that prehabilitation was initially looked at in the oncology field is with breast cancer.
Okay.
So there were some of those, and again, there's a unique high riskness to the head and neck cancer population that really make prehabilitation even more opportunistic in terms of impacting change and symptom burden and outcomes, because it's a different patient population.
Sure. Mm-hmm.
Yeah, this is a project that has been longstanding and dear to my heart, because it started when we identified really this population took a lot more of our staff energy, and we still were struggling with giving them the support that they needed in kind of a haphazard way, which is why we began this study to begin with, and then why we began this project to begin with, and then the DaCCoTA grant provided us an opportunity to really look at and study the effectiveness in a comparison way. It's been extremely humbling for me as a kind of a trench worker. I call myself. I'm not an academician. I am a trench worker, and it's really been. I don't know what I would have done without the team supporting me through this and dotting the I's and crossing the T's. And in the end, really anecdotally, we can say that administration of prehabilitation to our head and neck cancer patients has had a dramatic impact on outcomes, predominantly symptom burden, but also like just even the support that they get to be able to begin speech rehabilitation if they've had a laryngectomy earlier and things like that. Management of pain, etc. Anything else?
Well, if the mic is open, I do have another question. With head and neck cancer, do you know what sort of like in North Dakota compared to say the rest of the country or other parts of the country, how kind of it stacks up? Do we have more head and neck cancer, less, or about the same? Do you have any idea of the distribution?
I don't think I think we're kind of run of the mill. Dr. Cheryl would know that, or Rebecca Renfrew, our stats people that bring that in. I don't think we have a tremendously higher rate than other areas of the country. Yeah.
Okay. And then is the main risk factor, is that going to be something with smoking, or is that a different etiology, like more with the esophagus? Or do we even know what causes head and neck cancer?
Well, you know, we alcohol, tobacco,
okay, so that's a big one.
HPV. So there are certain things that causal rather than correlation. Is the debate. Do we know that
Sure.
causes or not? But chemical use. I know one of the first patients I had was a heavy duty snorter of different things and had nasal. Yeah, so it is associated with that, which is, you know, it's not always, but really it is what makes it more challenging, because when you have somebody with a heavy chemical dependency background, they have often the psychosocial
right.
issues that go along with that. Yeah. And it also, I might say, is what makes it extremely rewarding to be able to put a comprehensive program into place to help these people. That I forgot to mention that one of the things that is high risk is CD. So that's assessed in the prehabilitation program. And even though we don't have CD providers in Sanford proper, we did through the course of this development of this prehabilitation program, develop a resource for having patients evaluated. And if nothing else, we were much more attentive to, for example, people that were detoxing in post-surgically in ICU. Yeah, because we asked the questions and we knew what was going on. Yeah.
All right. Well, Dr. Hysjulien, thank you so much for your talk.
You're welcome. Thanks.
Yeah. So next up is Dr. Katti, and you can get started whenever you're ready.
All right. Thank you, Dr. Williamson. Good afternoon, everybody. The title of my presentation is Prostate Cancer Bone Metastasis Testbed for Regenerative Bone Therapies for Bone Metastasis. And that's the title of the grant from DaCCoTA. I'm Dinesh Katti from North Dakota State University. Next slide, please. So the participants, the students participating are Preetham Ravi, Gwyn Huang, Srinwanti Ghosh, Sharad Jaswankar, and Hanman Gaikwad. And the investigators include Dr. Parth Vyas and Dr. Sami Hashmat from Sanford Health, and Dr. Kalpana Katti and Dr. Jiha Kim from NDSU. Next slide. So although this is one project, I'm going to talk in, as three sub-projects within this project, and I will be talking about basically describing the objective, the background, some prior work, results, and summary for each of these sub-projects. Next slide. So here we, in the sub-project one, we create a patient-derived prostate cancer bone metastasis in vitro model. That is the first objective. Next slide. So the objectives here are isolation and characterization of prostate cancer cells from patient-derived tissue obtained from Sanford Hospital, culture patient-derived cells on 3D nanoclay bone mimetic scaffolds to recapitulate patient heterogeneity, and in general, validate bone metastasis in vitro model. Next slide. So in our group, for the first time, we have created a bone metastasis cancer testbed. This is done in our previous work. And this cancer testbed uses the tissue engineering approach where we have used nanoclays to build tissue engineering scaffolds. And these scaffolds are able to regenerate a human bone using human mesenchymal stem cells. And then on this regenerating bone, we introduce prostate cancer or breast cancer cells, and we're able to mimic the secondary tumor site or the bone metastasis site. And this is these are the scaffolds or test beds we are going to be using with patient-derived cell lines also. Next slide, please. So here is from the previous DaCCoTA grant, which was the PI was Dr. Kalpana Katti, and this was on patient-derived breast cancer cells. And here I'm just showing you two examples. One is the NT013, which is the hormone-positive breast cancer cells, and NT023, which is the triple-negative breast cancer cells. And what we see here is that the patient-derived breast cancer cells retain their tumor-related characteristics after dissociation. And also we compared these with the commercial cell lines, and we were able to show that these test beds are able to mimic both the triple negative as well as the hormone positive breast cancer, which we see in patients. Next slide. Also, using these 3D bone metastasis test beds, we're able to recreate the tumors at the bone metastasis site using the patient-derived cell lines. This is for breast cancer, which was previous work. Next slide, please. Also, in our previous work, what we had done is we're able to observe what happens to the bone in the proximity of the cancer tumors using these cancer test beds. And what we showed is that these prostate cancer tumors influence bone mineralization at the metastasis site. And not only bone mineralization, but also if we look at the bottom row, we see a significant disruption of the collagen fibril formation. So basically, we see differences in terms of the patterns, in terms of the various attributes of the collagen formation. So both the mineral and the collagen is affected, and we are able to show that on our cancer test beds. Next slide. So in this particular project, the first step is to obtain tissue samples. And so the first, what we did was we took tissue samples from the hospital immediately after surgery. And then the next step is the tissue preparation. Then the tissue was mechanically chopped with the scalpel. And then we had the enzymatic tissue dissociation, followed by co-culture of prostate cancer cells and feeder cells. And then from there, we're able to get the patient-derived prostate cancer cells. Once these patient-derived prostate cancer cells are isolated or developed, the next step is, of course, to put them on our 3D bone mimetic scaffold. So the steps for that include first the scaffolds themselves. Like I said before, these are the nanoclay-based scaffolds. These scaffolds are sterilized. And then we also have the human mesenchymal stem cells. And so the first step is to culture these human mesenchymal stem cells on the scaffolds. We wait for 23 days to obtain the bone-like ECM or the initial bone formation. And once that is done, we see the scaffolds with the cancer cells on this regenerating bone. And once that is done, we wait for 5, 10, 20 days period and observe the growth of tumors and do a whole bunch of experiments on those tumors grown at the bone metastasis site. Next slide, please. So for the prostate cancer tissue samples, we have obtained 8 samples up to date. We have 6 from the primary site, that is the prostate site. And we have been quite fortunate to get 2 from the bone metastasis site. So the 3003 and 3004 are from the bone metastasis site, not easy to get, but thanks to our collaborators at Sanford Health, we have been able to obtain those samples. Next slide, please. And so on the left-hand side, you see the cells from the primary side, the isolated cells from the primary side, and they show a epithelial morphology. On the right-hand side, the two are the 3003, 3004. These are from the bone metastatic side. These are showing more of a fibroblastic morphology. And so cell isolated from primary site exhibit more epithelial characteristics. Next slide, please. And so now the 3001 and 3002, basically this particular slide shows the extracted cells from the primary site. And so the primary site patient-derived prostate cancer cells express PSA, but are androgen receptor negative. So these are the characteristics of these two samples, 3001 and 3002 from the primary site. Next slide, please. And these are from the bone metastasis site, the 3003 and 3004. And here we see that the patient-derived prostate cancer cells from the bone site are PSA and AR negative. Now, let me mention here that these are just the extracted cells. So these have not been put on our 3D scaffolds. So these are extracted cells. And we are going to be repeating these experiments again, and we are also going to be doing some additional protein expression analysis to validate some of these results. So the patient-derived cell lines from bone site retain their aggressive behavior through expression of vimentin. Next slide, please. And so for this first sub-project, 8 tissue samples were obtained from patients with prostate cancer diagnosis from both primary site and bone microenvironment. From the 8 tissue samples, 8 cell lines were successfully established. The identified characteristics of lines 3001 to 3004. And in future, these cell lines will be cultured on the 3D bone mimetic scaffold. Actually, though, that work is actually going on. We don't have the results yet. Next slide, please. So the next one is a very interesting sub-project, which is isolation of patient-derived cells from bone chips. So during the process of obtaining tissue from the bone metastasis site, we also get bone chips. And so here we're talking about the isolation of patient-derived cells from the bone chips. Next slide. So here the objective is to isolate cells from tumor tissue and bone chips taken from bone marrow of patients with prostate cancer bone metastasis, characterize and classify these patient-derived bone stromal cells, culture patient-derived cells on 3D nanoclay bone mimetic scaffolds. Next slide, please. So we have been able to get the bone chips from just one sample, that is the 3004. Next. Next. And next slide, please. And so these are the cell isolation steps of bone chips from the prostate cancer tissue. First, we collect the tissue samples. The next one is the mechanical dissociation of tumor tissue and removing tissue from bone, followed by the enzymatic dissociation using shaking incubator. And then after that, there are several other steps. And eventually, we are able to isolate the cells from the bone chips. Next slide, please. And so here on the left-hand side panel, you see the bone chip, which is surrounded by the bone cells. And so the next shows the cells in the culture. So this gives you a better view of the cells in the culture. Next slide, please. So we wanted to see whether the cells which we have obtained from the bone chips, whether these are cancer cells or these are bone cells. And so here, the immunofluorescence results show lack of intensity of CK19, suggesting that these cells do not have cancer characteristics. So these are from the bone chips. And when we look at the nestin, it does indicate that these are likely progenitor or stem like properties these cells have. And we do see some fluorescence for OCN. So these do appear to be, these are from the bone chip, so these appear to be the progenitor bone cells. Next slide. So these patient-derived bone cells are displaying strong fluorescence intensity of nestin and weak intensity of OCN, suggesting these cells are likely stem cells or progenitor cells. Now, we are also going to do more work on classifying these cells.
2 minutes remaining.
Thank you. And additionally, one very interesting thing we are planning to do here is that we are going to use these bone cells to create a bone niche from the patient cells and put the patient's tumor cells on those. So we're trying to mimic as closely as possible the bone site, the bone metastasis site of 3004, the patient 3004. Next slide. The last one is the sandwich experiment. So using sandwich setup to evaluate the role of healthy bone on bone metastasis prostate cancer, and then evaluating new therapies using BMP's coated interlocking bone scaffolds. Next slide. So we have used these cancer test beds to study we found that the bone interface modulates drug resistance in breast cancer bone metastasis. We've seen that in our previous work. Next slide. We have also developed this BMP2, BMP7 coated interlocking scaffold, and these scaffolds, we have a patent on this, and these scaffolds are very efficient for non-union bone defects. Next slide. So in surgeries at the bone metastasis site, we create these bone defects because we are removing the cancer regions within the bone. And the idea here is to put these interlocking scaffolds to fill these bone defects. So here we want to study how the filling of these bone defects, whether it will have what kind of effect it would have on cancer metastasis, and whether introducing this rapidly growing bone is going to somehow inhibit the bone metastasis is one of the things. And we are also looking at using this for screening drugs for the bone metastasis site. Next slide.
All right. If you could,
All right.
summarize.
Thank you very much.
We'll have time
And
for
so
questions.
I will stop here. And next slide. I'd like to thank everybody for joining my presentation. And also, most importantly, would like to thank the patients for consenting to provide tissue. Thank you. I'm happy to answer any questions.
Dr. Katti, very good night to talk. I have a question. As long as I know that the prostate cancer cell, they also have a very developed system to crosstalk with the other tissue around, even nervous system, and the PNI is one of those things that initiated from the prostate cancer. So in your 3D model, is there any way also to look at this crosstalk system that this prostate metastatic cancer cell may have?
Yes, absolutely. And I think one of the things we are doing right now, although this is not part of the DaCCoTA project, it's part of the EPSCoR, is to look at co-cultures. So in addition to use the human mesenchymal stem cells and also using the cancer cells, we are also looking at a variety of co-cultures. Right now, we're looking at HUVEC cells and there are other cells. And so absolutely, we could be maybe having collaborations with others, especially in other areas like neural cells. I can see Kevin here and others, where we could look at co-cultures and see the effect. We are finding some very interesting crosstalk between the the bone cells and the cancer cells, and not only that, how it's changing the bone morphology surrounding the prostate cancer cells. So we are able to see both osteoblastic and osteoclastic responses on the bone on our test beds, depending upon the type of cancer cells, whether it is triple negative or MCF7. We are able to see different responses on these. So we are very hopeful that we'll be able to our results from co-culture studies could look at other aspects.
Thank you.
We are also putting these scaffold, these cancer test beds in fluid flow environment in bioreactors to mimic interstitial fluid flow. And we are seeing some very interesting results with that. We find that interstitial fluid flow increases cancer cell migration. We are able to take a close look at activation of, like, for example, the alpha V beta 3, for integrins. And so we are able to take a deep dive into what is going on at the cancer cells at the bone metastasis site. And recently, we've been able to identify some natural products which can inhibit cancer cell growth, at the same time not affect the bone cells. So we are able to see some of those exciting things with the cancer test beds.
It's very interesting you said that natural product. One of the tumorigenic proteins that it's also involved in prostate cancer is the heat shock protein mortalin, which is a HSP17 mitochondrial protein. So, and I know that prostate cancer also elevate this protein, especially metastatic one. Maybe I will be in contact with you to share because we have an inhibitor of this mortalin. So maybe we can.
Oh, absolutely. I would love to collaborate, and we have been collaborating with a variety of researchers, and would love to collaborate with you also.
Perfect. I will be in touch. Thanks.
All right. I'd like to thank Dr. Katti for his great talk. Next up is Dr. Roiko.
Hey, good afternoon, everybody. I'm going to switch gears a little bit and talk more about SARS-CoV-2 and other bacteria. So a little bit of a switch from cancer. Next slide, please. So as we all know, SARS-CoV-2 is a causative agent of COVID-19. It's caused a pandemic of significant morbidity and mortality over the past 3 years. Over these 3 years, there have been significant strides in understanding, preventing, and treating COVID-19. However, there are still several aspects of SARS-CoV-2 and COVID-19 that require ongoing and additional investigation. So in this project, we're looking at the relationship between SARS-CoV-2 viral variants, COVID-19 disease, and the upper respiratory microbiome, which is a site of initial infection. And the samples that were collected for this study are mostly collected from 2020 and 2021. So people that had sort of an initial infection with COVID-19. Next slide. So a little bit of background on the microbiome. We've probably heard this term a lot, and there's been a lot of research interest into the microbiome. It's the collection of all the microorganisms, including bacteria, fungi, and viruses that normally inhabit the body. The composition of organisms vary by bodily site and can fluctuate in response to environmental changes. You know, the microbiome is important, and it's increasingly understood to have an important role in health related to digesting food. Different bacteria can produce necessary vitamins, regulate the immune system, and protect against disease-causing organisms. So a lot of varied functions that we're still learning as we keep investigating the microbiome. Next slide. This study specifically looked at the bacterial microbiome, and we know that we're neglecting viruses and fungi when we look at this. However, there is a lot of variety with the bacteria that occupy each bodily site. And for those of us who are not bacterial taxonomists, I thought it would be a little bit helpful to include some background on what each of these different bacterial phyla compose of. So the main phyla that we find associated with the microbiome include the actinobacteria. These are gram-positive and have a high G+C content in their DNA. So examples of these bacteria include DaCCoTA, Crinibacterium, Cutibacterium, which was formerly known as Propionibacterium, and Gardnerella. If you look at these pie charts on this right figure, we can see that these bacteria are in the light blue, and they are really dominant in the hair, nostril, skin sites, and in the H. pylori negative stomach. The Firmicutes are also gram-positive, and they have a low G+C content. Examples of Firmicutes organisms include Clostridioides, Lactobacillus, Staphylococcus, and Streptococcus. In these pie charts, they're in red, and you can see there's high concentrations at various sites, including the vagina, oral cavity, esophagus, somewhat in the stomach, and they dominate also in the colon. The proteobacteria are gram-negative and LPS outer membrane. So this is also a very diverse phylum. Examples include Escherichia, Haemophilus, Helicobacter, and Moraxella. And the proteobacteria in these pie charts are in green. And we can see that they tend to be in the oral cavity and somewhat less in the GI tract. In the H. pylori stomach, you can see the green is overrepresentative because H. pylori dominates in that case. The Bacteroidetes are gram-negative non-spore-forming organisms. They can be aerobic and anaerobic. The examples here are mostly anaerobic Bacteroides, Phylobacterium, Porphyromonas, and Protella. And they are in the purple slice. You can see they are mostly in the GI tract. Cyanobacteria are gram-negative and can undergo photosynthesis. And this is most genera. There's recent evidence that there are some genera that belong to this phylum that are non-photosynthetic. So examples include Nostoc, Nyctococcus, and Melinobacteria. And these are in yellow, and we can see these genera show up in hair and nostril sites. And it's typically thought that these organisms, when they're present in the microbiome, represent environmental organisms. So you can imagine sites that are in contact with light and water might have these sort of environmental flora there, and their clinical significance is yet to be determined. Another phylum that we see often in microbiome is the Fusobacteria. These are gram-negative obligate anaerobes and often asaccharolytic, and this includes Fusobacterium, Leptotrichia, Snelia, and Streptobacillus. So you can see these in orange, and they are not a dominant part of any of these sites, however, they do have important functions. So I've included in parentheses here the newer names for these phyla. These were all renamed in about 2021. However, I will refer to these by their previous names just for familiarity. Next slide, please. So when you think about the microbiome and disease, there are some well-known cases where microbiome disruption is associated with disease. A well-known example is antibiotic treatment that disrupts the gastrointestinal tract microbiome and is a major risk factor for C. difficile colitis. The GI microbiome changes are also associated with other GI liver conditions such as irritable bowel disease, non-alcoholic fatty liver disease, and so on. Another well-known example is bacterial vaginitis, and that's associated with vaginal microbiome dysbiosis, so rather than infection with a particular microorganism. So for testing for vaginitis, we're really looking at the disruption in the microbiome, and there are various tests we do for that instead of culture for a specific organism. And it's important to note that disruption of the vaginal microbiome can affect the risk for HIV infection. Next slide, please. So when we're looking at the upper respiratory tract, we all know that this is in constant contact with the external environment via airflow. So a lot of environmental stimuli we're exposed to there, as well as viruses. So functions for the upper respiratory tract include filtering, warming, and humidifying inhaled air, olfactory sensation, and also innate and adaptive immune responses. The upper respiratory tract is a site of initial infection for many viruses, including SARS-CoV-2. However, the upper respiratory microbiome is really under-investigated, as indicated by PubMed search in June this year. So if we pulled up PubMed, I found 567 publications on the upper respiratory tract, and 55 of these were for SARS-CoV-2, compared to over 200,000 publications in total for SARS-CoV-2. And comparing 567, there's over 40,000 publications on the GI microbiome. So a lot of work that still can be done here. Next slide, please. So what's been previously published in the literature on the upper respiratory microbiome, we know that it changes across the lifespan and that there are shifts observed with other disease stages, such as otitis, lower respiratory tract infection, asthma development, and allergic rhinoconjunctivitis. The studies examining SARS-CoV-2 in the upper respiratory tract have produced varying results to date. So some early studies found no differences in the microbiome of SARS-CoV-2 positive versus negative samples, and others did. The differences all examined, these studies all looked at varying patient populations. Some were specific to outpatients versus inpatients, some ICU patients, some healthcare workers. So most of these studies are from very specific patient populations. So in this investigation, we collected a very large number of samples for over a year to further examine possible microbiome changes with SARS-CoV-2 detection. Next slide, please. And so specific aims for this study were to support public health epidemiology by doing SARS-CoV-2 whole genome sequencing to identify locally circulating strains. And then we wanted to look at COVID-19 outcomes for different viral variants. We also performed 16S rRNA gene sequencing to determine the microbiome composition of the upper respiratory tract samples. And then the goal is to examine the relationship between COVID-19 outcomes and differences in microbiome composition. Next slide, please. So whole genome sequencing was performed from remnant samples that were submitted for routine diagnostic testing. So we focused on nasopharyngeal swabs that were collected in viral universal transferrin media. And these samples were all tested in a CLIA laboratory by an FDA emergency use assay to detect SARS-CoV-2 viral RNA. Total nucleic acid extraction was performed via Promega kit and whole genome sequencing by the Arctic protocol, and whole genome sequencing results were submitted to GISAID with our last submission in October 2021, and viral variant calling by the GISAID system. Next slide. So in this cohort, we had 550 samples in which we identified a viral variant by SARS-CoV-2 whole genome sequencing. We found 29 variants total and 5 variants that were classified as variants of interest by the CDC at the time of identification. Predominant variants in this cohort include the B117 or alpha, B12, and various other variants that were not assigned a specific designation. Next slide, please. For microbiome sequencing in the bioinformatic workflow, we performed 16S metagenomic sequencing on the nanopore. This was all done by our colleagues at the University of North Dakota in the genomics core. FastQC for quality assessment, for CHOP for adapter contamination, removal, FastP to remove low quality reads, and Kraken2 microbial for analyzing bioinformatic pathways. So over 900 samples passed through the metagenomic analysis pipeline. Next slide. So the findings to date, the majority of phyla present in the sample are similar between SARS-CoV-2 positive and negative samples. And those are indicated in the bar graph there in various colors. So proteobacteria dominate, followed by Firmicutes, cyanobacteria, Bacteroides, and actinobacteria. However, we noticed a significant difference in SARS-CoV-2 detection, an increase in actinobacteria and a reduction in Firmicutes. So the actinobacteria are in that brownish color and the Firmicutes in the bluish greenish. Next slide. And then this is just a bar graph or dot plot of various genera, and most of these are not associated with any known human infections. So these tend to be more unknown bacteria that we're finding in these samples. And for SARS-CoV-2 positive samples, these are several genera and actinobacteria. You can see those are in brownish dots, and we have an interesting finding with specific spirochete in the blue dot that was really abundant. Next slide. Oops. There, oh, table. Next slide. There we go, thank you. So looking at actinobacteria versus Firmicutes, again, just a reminder, these are both gram-positive, various GC contents, and the notable genre for our actinobacteria are Actinomyces, Bifidobacterium, Nocardia, Mycobacterium, Streptomyces. And these bacteria can be pathogens, symbionts, and commensals. These bacteria tend to have extensive secondary metabolic pathways, and they produce about 2/3 of the naturally derived antibiotics that are in current clinical use. For Firmicutes, bacteria that are known to be in this phylum are Bacillus, Clostridium, Lactobacillus, Staphylococcus, and Streptococcus. Again, these can be pathogens, symbionts, and commensals. They can produce endospores, and they're often involved in fermentative processes. So we're seeing more of the actinobacteria and less of the Firmicutes and SARS-CoV-2 positive samples. Next slide, please. When we look specifically at ages, we do see the microbiome varying across the lifespan. So there are varying shifts in the phylum at different ages, and it kind of varies by age. For the very young patients, we see not very much difference in the actinobacteria shift in positive and negative samples at the N and the D. However, this becomes more pronounced in adult patients. We
2
see
minutes remaining.
Thank you. With the Firmicutes also across the lifespan. Next slide, please. So then looking at viral variants, we do see some differences in these viral variants. We performed this so far for the predominant viral variants with more than 30 samples. And for the B117, we again have reduced Firmicutes, and then for the B12, we have seen reduced actinobacteria. So that's a little bit interesting to follow up for further investigation. Next slide, please. We also looked at differences by gender and hospitalization status and found no significant difference. Next slide, please. So in summary, we have identified a respiratory microbiome shift in response to SARS-CoV-2 infection. We find the shift varies by age for both positive and negative samples and viral variants, but not by gender or hospitalization status. So in future analyses, we would like to compare COVID-19 severity for asymptomatic, mild, moderate, severe, and critical illness by viral variant and for microbiome composition. We'd like to examine the microbiome for organisms specifically associated with poor outcomes, and these could be used as possible prognostic indicators. And then we will look at microbiome for organisms observed with asymptomatic or mild infections, and these have a potential role as a probiotic. Next slide. So much of the sequencing and bioinformatic work is done by our collaborators at the University of North Dakota School of Medicine, especially Brett McGregor and Jungga Kerr. Additional support from Antrix Tiagi, Sergey Nechev, Bonnie Kumar, Sarah Steinquist, Mike Hill, Data Analysis Core, and other staff at Genomics Core. At Ultra Health System, we have support from Sarah Nasheen, our infectious diseases provider, the pathology laboratory services staff and research department staff, and we did a big shout out to the DaCCoTA Croft staff. Thank you. Happy to take any questions.
Well, I guess I have a bit of maybe a fun or less serious question. When it comes to the renaming of the bacteriophyla, where do you fall on that? What camp do you fall into? Are you sort of neutral? You like it? You dislike it? Where? How did that affect you?
It's hard to say. In the clinical microbiology laboratory, we have regulatory requirements to maintain current nomenclature. It is a bit of a a teaching and learning moment for everybody. We have to keep an eye on what's coming as far as name changes and make sure we educate everybody. Okay, this is now the name for this bacteria, or now we're calling this this. So that's an ongoing, I think, educational endeavor. Sometimes it is challenging. For example, several years ago, the organism used to be known as Clostridium difficile was changed to a different genus, and there was a big pushback from the clinical microbiology field to make sure it stayed C. diff and didn't become P. diff or H. diff or G. diff or something else. So it is now Clostridioides difficile, which is still close enough you can call it C. diff. So it's an ongoing discussion to make sure that we do apply new knowledge as needed to make sure we're classifying things appropriately, and then also to maintain the clinical significance of educating providers as to what bacteria we're dealing with. So it's an ongoing push and pull between old habits and new information.
Thank you. All right. Unless there are any other questions in the chat, QA, or just through a microphone, I think we can switch over to our next presenter. Thank you, Dr. Roiko. Next up is Dr. Nadeau.
Hello. So I'll be talking about my project, creating a tribally informed risk and protective factors survey utilizing an Indigenous mixed methods study approach. Next slide. So compared to white people, Native people have a higher incidence, and this is according to 2014 to 2018 data, have a higher incidence of lung, colorectal, kidney cancers, as well as liver, stomach, and cervical cancers that are caused by infectious agents. Next slide. Here is my concept model for cancer-related risk and protective factors. And the second here for the cancer-related indicators, the YRBS collects data from youth for these indicators, but unfortunately the data that's collected is not representative of tribal nations. The YRBS tool is really meant to get a national snapshot, and it does a great job of that, but unfortunately it's not, doesn't do a good job with tribal communities. And so we know just from previous research that the behavior that happens with the youth leads to cancer over the lifespan. And so we know that substance use leads to cancer over the lifespan. We know that diet and physical health related indicators lead to cancer over the lifespan. And we know that sexual health behaviors impact cancer over the lifespan. What we don't know is the risk and protective factor profile for these cancer-related indicators that lead to cancer over the lifespan and ultimately result in cancer-related morbidity and mortality. And so my project is specific with the Native community and my tribe, specifically the Turtle Mountain Band of Chippewa Indians, and I've been working on this project with them for the last few years. Next slide, please. And so the goal is to determine the prevalence of community-level protective and cancer-related risk factors for tribal youth by creating and administering this community-informed data collection tool. Next slide. And so currently, a tool that assesses cancer-related risk and protective factors for Native youth does not exist. There are a multitude of factors that can impact youth's health and well-being and can lead to cancer later in life. So understanding the risk and protective factors and the social ecological presentation of these factors at the tribal level can inform community efforts. Next slide. So mixed studies process. So this is an exploratory sequential mixed methods design. And I conducted and published a mixed studies review. And then during that time, and during COVID, things kind of slowed down, as we all know. And so I kind of shifted into a different phase of my research and just scanned the nation for what I could find. Well, I should say Turtle Island, for what I could find for data collection tools that had been administered in tribal communities with the youth, just kind of building this potential question repository that could be looked to help inform the eventual creation of this data collection tool. And currently I'm conducting the qualitative phase of my study. And then once this phase, we're almost done with data collection. Our data's been transcribed. We're creating our themes. And so, yeah, so it shouldn't be long before that's done. Then we're going to create a community report and report that data back. But that data is also going to be used to inform the creation of the data collection tool. Next slide. And so this is the publication that came out on the Mixed Studies Review, Identification of Cancer-Related Risk and Protective Factors for American Indian Youth, a Mixed Studies Review. First Mixed Studies Review of its kind that's ever been put out there. So we found a lot of interesting findings from this work, and it's leading to other initiatives as a result. Next slide. So there's a lot of things that are in this, but I just touch on a few things because only got a few minutes to talk. But so substance use, some of the key results, we've seen that substance use, this key cancer related indicator with the presence of associated risk and protective factors occurred at all levels of the socio-ecological model. So basically what I did for this mixed studies review was I gathered systematically, looking at the literature, including qualitative, quantitative, and mixed studies literature, to see research that had looked at risk and protective factors for cancer-related indicators, across the nation. And so what I ended up finding when I did this, and how I organized it was, I said, okay, what are we seeing at the individual level? What are we seeing at the interpersonal level, the community level, the policy level, and probably most importantly, the cultural level, because cultural level, we know from previous research that cultural level impacts all levels of the social ecological model. So by investing in culture and creating strengths-based and community-rich strength environments, we can improve the health and well-being of our tribal communities. And so in relation to substance use, we could see that this, the presentation of the risk and protective factors span all levels of the social ecological model. So that was an interesting finding that for the research that's been done, it actually presented across the social ecological model. So efforts to reduce substance use related risk and protective factors should be aimed across all levels of the social ecological model. Substance use was also a risk factor for sexual health indicators, and it was a risk factor for physical health, diet indicators, and both at both the individual level. Next slide, please. So this was the first of its kind, like I said. Protective factors, they span multiple levels of the social ecological models, and then findings from this mixed studies review can guide strengths-based health promotion and prevention programming. And then I'm also going to use it to inform the creation of my data collection tool. Next slide. So the qualitative phase. So this has been kind of a fun part, because I went out into the community and I said, okay, this is what I did for my mixed studies review. And I kind of had an idea of what ideas for them to consider. I'll put it that way. And I said, I was thinking we could do focus groups with the youth, and, we could do it with females, males, non-identifying, and then also females and males that were in school and out of school, and then non-identifying in and out of school. And I was going to do key informant interviews with adults who work with youth. And then when I sought community input, they're like, no, we don't want you to do focus groups with the youth. We want you to do it with cultural knowledge bearers, with administrators, with behavioral health specialists, and then key informant, then have key informant interviews with the youth, and then with other adults that can't make these talks. And I said, okay, so they totally flipped it, totally informed the development of my process for doing these interviews. And then data from these interviews will be used to inform the data collection tool that I'll be developing, and then also to, giving back to the community to inform their current efforts. Next slide. So this is just a breakdown of where we're at. 21 youth surveys, 6 adult interviews with cultural knowledge keepers, 1 behavioral health focus group, and 1 high school administration focus group. Next slide. So the preliminary findings is we see as well that culture influences health, that adults see a lot of opportunities for connection to culture, but unfortunately the youth see it as optional with limited access to culture. Next slide. Preliminary findings also showed that there's an overall lack of education. Sexual health is not offered in our community, unfortunately, which is very unfortunate because we know that that leads to cancer over the lifespan. And the really sad thing that I'm seeing is that it's not something they can go home and learn, because now we're into like generational effects of like, well, the parents didn't get it in school either. And so that's coming out in our data, that we're seeing generational lacks, like generational impacts from lack of education. How to live in a good way. There's people in the community said that there's a lot of focus on academia and like getting ready for tests and doing great in school, and like kids are stressing about passing this test or passing that test, but there's not a lot of time spent on like how to live in a good way and like independent living skills. I had that when I was in high school in Delport, but they don't offer that anymore. So it's been, and the classes that are offered are optional, and so they're just not getting these skills that are needed for like everyday living. Next slide. So future directions: analyze and disseminate the data of the qualitative findings, create this community-informed survey, validate the community-informed survey, and then administer the community-informed survey. And then the hope is once this data comes back, that we can bring it to the community and the community can use it to inform and drive their intervention efforts with the youth. Next slide, sorry. And I guess I'll just say that I'd like to thank the students that work with me. I had a medical student that worked with me, a couple of masters of public health students, and then Dr. Tyler Persian. He works with me in Turtle Mountain, and I work, to build capacity at the tribal level so that this work is sustainable. And yeah, I'm looking forward to continuing this work. That's all I have. Thank you.
All right, we'll open this up for questions now. Not hearing any questions. Let's thank our presenter, Dr. Nadeau, again, and we can shift to our final presenter of the session, Dr. Sun. You can, start presenting whenever you're ready.
Thank you so much for the introduction. Today I want to share some update for exosomal collective attribute for non-invasive cancer screening. Can you hear me clearly? Okay, great. So I'm going to start with the introductory to the exosome. Exosome is kind of a small vesicles secreted by the cells, goes into the circulating system containing some information from the cells. So it's small vesicles size from 100 to 200 nanometers in diameter, and a lot of molecules actually contains in the small vesicles we can use to, for example, detection or even treat the cancer cells. Can you go to the next slide? Okay, so because it's really protected environment, membrane protected environment inside the exosomes, so the molecules inside the exosomes kind of stable. So clinicians use this, many researchers use this property to dig into the biomarker inside the exosomes, use the exosome as a biomarker reservoir. So till now, a lot of individual biomarker has been developed, but unluckily, none of them are actually actively used in clinical applications. So the problem is, can you turn to the next slide? The problem is none of the individual biomarker can indicate tumor status. So we actually did a protein-protein interaction study. We found that tumor cells has more complex interaction, protein interaction. That means once you use one single marker, it's not possible to use one single marker to indicate the whole status of the cells because they are very heavily dependent to each other. So that's why we consider collective attribute, not a single marker, collective attribute of exosomes as a non-invasive marker for cancer detection. So please, next slide. So I'm going to update you 3 collective attributes we developed in our lab. The first one is beta sheet rich of exosomal protein inside exosome for cancer screening purpose. Can you go to the next slide? So this one we published as journal cover, and it's the first study of this kind. And I want to tell you a story about how we developed this. Please, next slide. So we actually found the phenomena from CD measurement, circular diagram measurement. We found there's specific patterning of tumor exosome and the tumor cells for CD measurement. We found this patterning is closely related to the beta sheet rich of tumor cells. So then we hypothesized, please go to the next slide. We hypothesized that normal exosome from normal cells will have alpha helix rich, but the tumor exosome from tumor cell will be beta sheet rich. So in order to validate this hypothesis, we use several different experimental methods. Please go to the next slide. So, oh, can you see it? Okay. Oh, I think the image is not clear. But we use several experimental methods. CD is one of the experimental methods, and then we use FTIR to validate the same phenomena. We found a beta sheet rich of tumor exosome and tumor cells. We also use fluorescent staining. We use a specific fluorophore to target at the beta sheet proteins and then label the beta sheet protein. We found the same thing, beta sheet richness of tumorous exosomes. So those are the experimental methods we use, counting the protein as a whole inside exosomes. So please go to the next slide. Next things we did is we want to identify and then count in the individual proteins contribution to beta sheet rich of tumor cells. So we did some proteomics study, and then combined with bioinformatics and also simulation, we found a proof counting the individual proteins contribution that the beta sheet rich is true inside exosome from tumor cells. Okay, this research was published, and also we developed the tools to analyze it. You can use it. It's called FAPS. It's a website you can use. I actually had a barcode you can use that to access this website. Okay, please go to the next page. Okay, so we found beta sheet rich of tumorous exosomes, and then how we can use it in clinical settings. So we developed a clinical assay. It's called ExoIP-THT, combining immunoprecipitation with fluorescence staining. So with this method, we can differentiate the pancreatic cancer from pancreatitis, which is a disease, not cancer disease, and then also healthy control. The performance of this assay actually comparable to CA19-9, and also it's complementary to CA19-9. We did a correlation study. It didn't show correlation to CA19-9. That means it's complementary. You can use this method as a complementary to the CA19-9 to better diagnose pancreatic cancer. CA19-9, which is commonly used clinical marker for pancreatic cancer. Please go to the next page. Okay, another collective attribute I want to introduce is the exosomal DNA to protein ratio. We call it ExoID. Please go to the next one. So this collective attribute we initially found it from spectrometer. We found that there's a spectrum difference between tumor exosome and normal exosomes. And then this pattern difference, how we can indicate this pattern difference to more friendly to clinical operator. So we define, please go to next page. We define a ratio, a absorption of 260 and an absorption of 280. This ratio, we call it nuclear to protein ratio. We found this ratio for each type of exosome from different type of cells. They have a unique value. They have a unique value. That make we name this specific type of marker ExoID, because each exosome from different type of cells will have a different value. And then we also find, please go to the next page. We also find this value actually is higher in tumor cells. That means exosome from tumor cells is higher in ExoID value. And we've also found a rationale for that, because of the DNA inside exosome is actually higher in tumor cells, in tumor exosomes. That's the reason why this ExoID value is higher in tumor exosomes. Please go next page. So we also work on the rationale why this ExoID value is higher in tumor exosomes. We know that only 2 sources of DNA inside the cells is from either mitochondria or nuclei. Since the cell is not dying, so the only rationale from the only possible source in the exosome for the DNA source is from mitochondria. So next, we want to validate this. Please go to the next page. Oh, still. So we actually validate by using the engineered knockdown method. We found that mitochondria actually is determining the DNA contents inside exosomes. Please go to next slide. So actually confirm that mitochondria DNA contribute most in high ExoID value inside the tumor exosomes. So this is our conclusion for this publication. Can you go to the next slide? We found ExoID value high in tumor exosomes. Then the next question is how we can use in clinical settings. Similarly, we developed a clinical-oriented assay that can be used to distinguish healthy and then pancreatic cancer cases. So as you can see that combining CA19-9 with the ExoID method together will have a best performance of distinguish. So we then suggest that we use the CA19-9 complementary to the ExoID value method. Please go next slide. That's a summary of ExoID project. It's published in Biosensor and then Bioelectronics. Please go next slide. The third collective attribute I want to introduce is exosomal mitochondrial DNA. Please go next slide. Yeah, from the previous studies, we know that the mitochondrial DNA contribute most inside the exosomal DNA. So can we actually use that mitochondrial DNA rich inside the exosomes to distinguish, to diagnose pancreatic cancer? So with this in mind, we developed this project. Okay, next, please. We designed an assay using mitochondrial DNA, using the immunoprecipitation, and then also use PCR, quantitative PCR. We have validated this in vitro and also in vivo. We found this assay is not correlated to the tumor growth. That means you can use this assay to distinguish for cancer screening purpose, not a prognosis purpose. Okay. Please go next slide. Okay. So we also perform some clinical samples validations. We found this assay can distinguish a healthy pancreatitis and PDAC, pancreatic cancer. I want to emphasize the beauty of this method. This method uses PCR as a detection, quantitative PCR as a detection, but this method uses duplex probes. That means you can target the mitochondrial richness using the duplex probes at the same run. That's saving the time and also give less crosstalk between probes. You can use multiple probes in the qPCR, quantitative PCR, but when you use multiple ones, it will introduce more crosstalk between the fluorophore. So that's why we use duplex to minimize the crosstalk. And also in this method, we use DNA isolation-free method for qPCR. This saves a lot of time because if you're doing qPCR, the most of the time you are dealing with DNA isolation for quantitative purpose. But this method we specifically designed with no DNA isolation, which make this method more clinical oriented. And then
2 minutes remaining.
Okay, I'm done with that. So this method, the feature of the method also included less sample consumption and then less time consumption. You can perform this assay within 1.5 hours. Okay, and also high throughput. You can use this method as a high throughput method for screening purpose, cancer screening purpose. Okay, please go next slide. So this three collective attributes we actually found in our lab are our main research funded by DaCCoTA. So please go next slide. I want to thank DaCCoTA for funding us and then make those exciting research possible to the public. And I want to thank all my students who work with me. They are excellent students and then the really motivated students. I also want to thank all the collaborators in our university, also in the University of DaCCoTA. Thank you.
All right, we have time now for questions.
Dr. Sun, very nice talk. I really enjoyed.
Thank you.
What about the mitochondrial protein, especially nuclear protein that end up in mitochondria?
That's really nice questions. We actually did some research on mitochondrial proteins. We found actually mitochondrial proteins are beta sheet rich. That's for sure. We had that in our paper, that data in our paper too. So the question is how we can use that mitochondrial beta sheet rich proteins inside the detection or inside the treatment. So that's the question that we want to address from now on.
just as a curiosity, I don't know, you did a detailed proteomic. Have you seen any heat shock protein in your exosome?
Which protein?
Heat shock protein in HSP 1790.
I think so. Then there's plenty. I think I saw that in the data. There's plenty of them. Yeah, but we didn't focusing on the one single protein. We are focusing on the collective attribute. So we actually take for example, we use beta sheet. Each of the protein will have a beta sheet. We summarize all the beta sheet percentage from each individual protein and then summarize it to have a collective attribute of a protein. So individual protein, we're not too much focusing, but of course I saw that proteins in the proteomics list.
Okay. And finally, one of your results that you initially used the nanodrop and the different pattern of the protein distribution in the exosome from tumor versus exosome from the normal.
Yeah.
Very interestingly, we had a similar kind of pattern when we use the gradient fractionation to see the protein between normal and cancer cells.
Yeah.
In colon cancer, and we saw it was amazing that I exactly got similar to what your result looks. For the tumor is basically 2 peak of the proteins. Yeah.
That's great. That's great. Actually, that's great to hear. That would be a kind of encourage for us to go further with this.
Yeah, it was very interesting. I really amazed that I see that exactly. It's very similar to what we got it. Yeah.
Great. That's great to know.
I definitely will be in touch with you with my data to see further if we can work together in the future.
Sure. Thank you so much. Thank you so much. I'm looking for it.
Thank you.
Any other questions? Okay, if not, let's thank our final presenter, Dr. Sun. And with that, that ends the main portions of our symposium today. So thanks for all the awardees, all the presenters, everyone who talked. Hopefully we had a very enlightening and collaborative experience here. So the last thing we have here is just the closing and wrap up. Dr. Schwartz, would you like to say a few words?
Very few. The few that I do want to say is thank you. I've really enjoyed this. This has been actually a great day for me. I've had a chance to hear a bunch of things that I wanted to know more about. And also when I stepped out, I had the opportunity to talk with the external advisors for the DaCCoTA who were very, very complimentary and really helpful. So I want to really extend a heartfelt thanks to the presenters and the poster makers. And again, to thank Mark Williamson, Nikki Voigt, Sue Thompson, Kent, Ripplinger, Miranda, Lighthouser, Jamie Scholl, Savannah Macias-Dougherty, Shane Samuels, and everyone who was involved in putting together what I think was our most successful Zoomathon. It's very hard to put these things together. And I really was delighted to hear from all of you. So thank you so much. And thank you for lasting the whole day. You guys are the endurance champions. But I have to say for my own self, I wasn't getting Zoom fatigue. I thought that the presentations were so interesting and so well timed that I didn't consider it fatiguing at all. It was really great. Thank you so much. Again, there's very good documentation of this through the DaCCoTA program booklet. So I would encourage you to contact anyone whose talk you found interesting or whose posters you found engaging, because the whole point of this really is to mix it up. So to get as many people working together as possible. Thank you again to all the participants. And I look forward to hearing from you if you have suggestions about how to do this better next time. Let me know, and we'll do what we can to work on continuous improvement. But thank you to all the presenters and to all the people who made this possible. Have a great weekend.